This is a production of Cornell University.
Our guest this afternoon is Professor Norman Uphoff. He is a professor of government and international agriculture and currently serves as director of graduate studies for the Cornell Institute for Public Affairs. He has been on the faculty of Cornell University since completing his PhD in political science at the University of California, Berkeley in 1970. And for most of his 20 years at Cornell, he has chaired the Interdisciplinary Rural Development Committee based in the Einaudi Center for International Studies. Since 1990, Uphoff was selected as the first director of the Cornell International Institute for Food, Agriculture, and Development, CIFAD, and he served in that capacity for 15 years. During this time, he became acquainted, starting in 1993, with the system of rice intensification in Madagascar, which will be the subject of the talk today. After retiring as director of CIFAD in 2005, Uphoff joined CIFA as a core faculty member, having himself completed Princeton University's MPA program at the Woodrow Wilson School in 1966, before undertaking his PhD at Berkeley. Professor Uphoff's focus as a social scientist working on development challenges and opportunities in Africa, Asia, and Latin America was on participatory approaches to agriculture and rural development, with a focus on farmer organization, local government, decentralization, social capital, related subjects with particular interest in management of natural resources. From 1978 to 1982, he served as director of the USAID-funded Rural Development Participation Project. Then from 1982 to 1988, he was a member of USAID's Research Advisory Committee. He served as a consultant on participatory agricultural and rural development for USAID, the World Bank, the United Nations, the Ford Foundation, the African Development Foundation, the Asian Development Bank, the Asian Productivity Organization, CARE, Community Aid Abroad, and other agencies. His books include Local Organizations, Intermediaries in Rural Development with Milton Eastman, Improving International Irrigation Management with Farmer Participation, Getting the Process Right, Local Institutional Development, an Analytical Sourcebook with Cases, Puzzles of Productivity in Public Organizations, Learning from Gail Oya, Possibilities for Participatory Development in Post-Newtonian Social Science, Reasons for Hope, Instructive Experiences in Rural Development with Anirudh Krishna and Milton Eastman, and Reasons for Success, Learning from Instructive Experiences in Rural Development, also with Milton Eastman and Anirudh Krishna. Since 2000, Professor Uphoff's work has been increasingly focused on agroecology and sustainable development, prompted by his work with the system of rice intensification. He has now written a number of articles in the agriculture science literature, and his most recent book, Biological Approaches to Sustainable Soil Systems, an edited volume with 102 contributors from 28 countries, was published by CRC Press in 2006. He thinks that after hearing him talk about the system of rice intensification and what it has to offer for more productivity in the sustainable agriculture sector in the 21st century, you will understand why he's become so absorbed in agroeconomy, soil biology, and soil ecology. Please welcome Professor Norman Uphoff.
Thank you. I'm going to speak from in front, if I may, because I can move a bit, and also I can see better up to the screen way up there. What's wrong with this title? Meeting world food needs by raising the productivity of land and labor and capital and water. That should shock you. Can it be possible to raise the productivity of 4 factors of production all at the same time? You've all done your Econ 101 where they told you that you can't do things like that. After all, aren't there supposed to be trade-offs? You know, is there such a thing as a free lunch? You're supposed to answer no. I'm going to tell you just maybe we can have a yes answer to that, but those are very valid questions, and so maybe in the question period you'll want to come back to this. The challenge for the 21st century is how are we going to be able to produce more with less? This sounds impossible, of course, but if we think about it, if we're going to have sustainable development with a higher population, higher demands for income, producing more with more is going to be very, very difficult. It's like squaring the circle, so to speak. And also, if we're going to be trying to promote equitable development, if we have this kind of development path which is always more, I think we're not going to be able to make it. So anyway, can this be accomplished? Isn't this something like the mythical perpetual motion machine? That should be sort of going through your heads. And I'm going to suggest that in the realm of biology, which is different from the world of engineering and other disciplines, that this may be possible. In 21st century agriculture, we cannot, I think, continue to do more of the same. Things that worked very well in the 20th century, for a variety of reasons, aren't going to work as well in this century. Our arable land per capita is reducing. Our population, of course, goes up each year, so the amount per person comes down, particularly because we're losing productive land area to urban expansion, and because so much of our land is becoming degraded because of erosion, salinity, and other kinds of causes. Water supply for agriculture is declining because of the competing demands for domestic use and industry, and because climate change is reducing both the amount and the reliability of water supply. So the kind of agriculture we practiced in the 20th century, very large areas, mechanized, extensive cultivation, become less feasible when we have more population, less land per capita, less water, and less reliable water. And also pests and diseases are likely to increase with climate change because as temperature goes up, many of these buggies that we don't like become more and more severe. Energy prices will surely be higher in the 21st century than the 20th century, affecting both our production costs and our transportation costs. The model we had in the 20th century of having high inputs with subsidized energy costs, which meant also lower fertilizer costs, lesser pesticide or herbicide, other costs, isn't going to work. And the idea that we produce food here and send it halfway around the world somewhere else because the price is lower changes when you start having 2 and 3 times more costs of transportation with higher energy costs. So I think that there's a lot of reasons to expect that the 21st century is going to need a different kind of agriculture than we benefited from the 20th century. Our environmental impacts are a bigger concern. We're not as willing now, or maybe even as able, to continue polluting our groundwater supplies, affecting biodiversity and so forth with our agricultural interventions. And we also want access to technology to be very broadly available, not limited to the larger and richer producers, because poverty is continuing to grow in spite of all our efforts from the 20th century. So many of the world's poor, as we know, were bypassed by the Green Revolution. And also food quality is becoming an issue. It isn't just that people want organic food because it tastes better, because we're trying to have chemical loads and loadings in our food supply that's getting really very worrisome. So anyway, I'm talking about SRI today because most of you are not agriculturalists, but I think you are interested in the kind of world that we're going to have in this century. And I'm saying that SRI points out a path for what I'm calling postmodern agriculture. You probably haven't thought about that concept before. You know about postmodernism in various areas, but we've known what modern agriculture was in the 20th century. And I suggest we need a postmodern agriculture, which will in fact take into account these various trends and forces which I was just describing briefly and refashion the way we produce our food. The comparative advantage is declining for large-scale, mechanized, monocropped production where we have this long-distance trade. That's not a matter of opinion, not a matter of ideology, it's a matter of fact. And so we need to find ways to reduce our energy intensity of production. And consumer environmental demands are for growing healthier products. So we have to reduce some of our chemical dependence, which is a nice way of talking about, inorganic agriculture. And postmodern agriculture must be the most modern agriculture, meaning it's science-based. This is where it departs from typical postmodernism, which is anti-science in the humanities and social life. For agriculture, it's got to be very scientifically based, because if we get it wrong, there's a price to be paid. Now, the system of rice intensification I'm going to talk about was developed in Madagascar after 20 years of observation and experimentation by a really remarkable man, Father Henri de Launier, a Frenchman who went to Madagascar in 1961, sent by the Jesuit order to be an agricultural advisor. Happily, he had trained in agriculture at one of the best French schools before he went to seminary. So he did know some agriculture, but he didn't know rice, because don't do much rice growing in France. And he spent 20 years just observing, testing, and trying, and he synthesized a set of practices. 1983-84 is the year we date it as, where how do you manage the plants, the soil, and the water and the nutrients differently and more productively, creating a more beneficial environment for the rice plants to grow in. For plants, we use very young, tiny seedlings, 8, 10, 12 days old instead of 3, 4, 5, 6 weeks old, and then transplant them very carefully. Instead of plunging them into the ground, which people do all around the world, if you plunge them in the ground, the root tip comes up, so you have like a J. And it takes a week or two for the plant to reorient its root downward and start growing again. Where if you slip them in sort of sideways, you get an L shape. The roots don't have any shock, they don't stop growing, and the plant's very vigorous and healthy, and much wider spacing. We cut the plant population by 80 to 90%. You get higher yields, which is a fraction of the seed and a fraction of the plants. The soil, we actively aerate it with a little weeder during the growth period to stimulate the soil biota. We have no continuous flooding of the soil, saving a lot of water thereby. And the nutrients rely on compost rather than fertilizer. You can use fertilizer with these other methods. It helps. But the very best results come when you use purely organic methods of fertilization. This is a picture of Father Launier shortly before he died in 1995, making a field visit. This is the field of rice in Madagascar. This is one of the better ones. I won't say this is typical, but look at those rice plants. They're tall. I mean, those Malagasi's aren't as tall as I am, but they're reasonably tall. It's up over their heads. And you see those panicles of rice? If any of you have ever had any experience with rice, those plants should be falling over with that heavy head. But no, they're upright, they're strong, vigorous. The system was initially developed or promoted by an NGO that we worked with, Association Tepicina, which Father Launier founded with Malagasy friends in 1990. We've been evaluating it since about 2000 and trying to work through an international network, which CFAD, Institute I was director of for 15 years, began working with. I'll just mention briefly, that you may want to come back to this in the question period, we've had no significant donor support, though it's starting now. We've had some resistance from the scientific community, even here at Cornell, and been relying mostly on civil society for the spread. So there's a whole story about this in terms of civil society. In 1999, there was only one country in the world where it was even known about, Madagascar. Today, we are up to 32 countries where we've been able to demonstrate these methods give you a healthier, more productive plant. You see the range of countries. I don't have Egypt and Rwanda colored in yet, but they have now reported in with their results recently. That's a picture in China. I was taken on a visit by the China National Rice Research Institute to Tiantai County, where they were introducing SRI. And I love to see it in Chinese and SRI. This is research done at the China National Rice Research Institute, where they took the same variety and simply changed the management. And the purple is what you want to look at, because panicle means the grains. And this is different stages after the plant flowers: initial heading, full heading, milky rice stage, waxy rice, yellow rice stage. And you see how at all stages, all the different plant organs are bigger and heavier, but particularly the grain just mushrooms out when it's been given this different kind of management compared to the conventional rice, which you see on the right. The China National Rice Research Institute did 2 years of trials with comparing what they call standard rice management with new rice management. They didn't call it SRI because it's not quite SRI. It's about 75% SRI. But the standard was like 30-day-old seedlings versus 20 days, 20 by 20 spacing versus wider spacing, 30 by 30, continuous flooding, which is the norm for almost all rice production, if you can, versus alternate wetting and drying of the fields, and then 100% fertilization through chemical amendments versus 50-50. They didn't go all the way like I would recommend, but 50-50. And this is the results of different plant populations. You see, at 150,000 plants per hectare, or 180,000 plants, or 210,000 plants, if you do the standard rice management, it goes up, 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 up. Not a lot, but as you put more plants into the field, you get some higher yield. That's what you expect with standard production. If you do these other methods, young seedlings, wide spacing, no flooding, you get the opposite trend. In fact, at all densities, you get more yield, and you get 2.5 tons more yield with less water, less fertilizer, less seeds. This is based on 2 years of results done by the Chinese Rice Institute, the Chinese IRI, and with 2 different varieties, with all the standard scientific methods. So what you do is you get a different phenotype, a different plant type when you change the growing environment of the plant. So fewer plants, less water, less fertilizer gives you more yield rather than less, which you would expect. Anyway, this is from Indonesia, a picture from Lombok. 2 plants, same variety, same age, and you see the difference in the way the plants have grown in that time. This is results from 9 seasons of evaluation done by the Japanese private consulting firm Nippon Koi. And there were 12,133 trials on farm comparing conventional methods and SRI methods. Total area was 9,429.1 hectares. They have on every single one of those the data, the measurements. Average increase in yield was 3.3 tons per hectare, 78%, with 40% less water, 50% less fertilizer. Labor, they said it was essentially neutral, though some farmers are saving some of it more. On average, no more labor, and cost of production went down by 20%. And the net income on some of the farms, they went out and did the measurement of yield and then income, costs, and so forth, that the SRI methods gave 6.2 million rupees per hectare versus 1.2 million with the usual cultivation, where farmers have heavy costs of inputs, not a lot of output. If you reduce the costs and raise the output, the margin, the profitability goes up by multiples. This is a picture of the team leader for Nippon Koi in his office in Jakarta, showing the dried plants. So you see both the plant, those are single plants on the left, and look at the roots, you see, compared to those 2. So we get a very different plant type. This is in Cambodia. This is, again, one single plant from one single seed. And I met this woman about 6 months after we got this picture, and I thanked her and said we were using this picture in PowerPoints and on the cover of a book published in Germany. And she looked a little worried when I told her that. And I wondered if we'd broken some taboo about having married women, being shown all over the world or something. So I said, what's the problem? She said, she wasn't boasting. She said, if I'd known what you were going to do with that picture, I would have gotten my biggest one. So this is one, as far as she was concerned, just a random plant from her field. This I love. This is a report from the Latter-day Saints Charities in Cambodia, where they learned about this method from our NGO partner. And farmers have been getting 1 ton yield, because it's no irrigation, just rain-fed agriculture. 146 farmers averaged 4 tons yield first season by changing their management of the plants, the soil, the water, the nutrients. And the nice story is these 3 boys transplanted their father's fields in 1 day with almost a hectare. And this farmer went from 1.2 to 5 ton yield. He had 4 times more yield. And this was his neighbor, with all these people out there spending a whole day, 3 or 4 times more labor to transplant, and yet they still got less yield. So the fact that you can have, some boys go out and just do these methods real quickly and get a 4 times greater yield is an incredible benefit for some very poor people, because when you're getting 1.06 tons per hectare, you're really in a poverty situation. This is Sri Lanka, showing 2 fields, same variety, same soil, and same drought. These fields haven't been watered for 3 weeks, and you see the one on the left, which was flooded continuously during its early stages. So the roots degenerated from no oxygen, suffocation. Whereas on the right, SRI meant you give them just a small amount of water periodically. So the roots went deep, stayed healthy, and it's withstanding the drought quite well, thank you. This is from Vietnam, about an hour north of Hanoi, after typhoon. The woman's holding up an SRI plant on the left and a normal plant on the right, and behind her is the SRI field and a normal field after a typhoon went through and just blew all the, that's a picture without the woman standing in front, so you can see the effect that these plants can withstand the stress of storms. The Vietnam National Integrated Pest Management Program did a study in 2005, 2006 in 8 different provinces, side-by-side fields, farmers' fields, comparing the incidence of pests. The major diseases were sheath blight and leaf blight, small leaf folder, brown plant hopper. And in the spring season, there was 55% less incidence of the major pests and diseases. In the summer, it was even 70% less, just because of the natural resistance that the plants could have when they were grown with wide spacing, good soil aeration, organic matter, and so forth. In India now, we're starting to see the spread of SRI rapidly, and this is our best country, Shailesh, for SRI these days. Tamil Nadu state began in 2000. By 2004, they're up to 40,000. Last year, up to 430,000 hectares. This year, they said 750,000 hectares. That's what? That's about over almost 2 million acres. Yeah. And the average yield this year was estimated to be 7.5 tons per hectare for the whole state, versus 5.4 as a previous record. So that's almost 2 tons added yield with less water, less seed, less cost. In fact, even less labor, according to the Minister of Agriculture. Himachal Pradesh and Uttarakhand, up in the Himalayan foothills in the north of India, the NGO doing this work had only 40 farmers in 2006, 597 the next year. This year, 12,042, 20 times more. Because now they're getting WWF funding and Tata Trust funding. Tripura State, one of our best cases, they had 44 farmers in 2002. 3 years later, up to 880, and the Prime Minister or Chief Minister of the state and the Ag Minister and the Finance Minister came and actually saw the fields, said, okay, we'll give you a third of the state budget for agriculture to spread this. And there's a remarkable person, I'll show you his picture in a minute, who took this on as a task from 880 to 32,000 to 72,000 to 162,000 this year. This is a field in Tamil Nadu. Just look at the rice grains on that. It's like a carpet of grains. This is an organic farmer who's doing really well with these methods. This is the agriculture research. This is a state sector research. A lot of our work's been done by NGOs. But this fellow is a government employee. He got a copy of my paper in 2000 that had been photocopied so many times he said he had to take a pencil and fill in some of the letters and words to read it. But he did it. And he tried it himself in 2000, 2001. 2002, he got 44 farmers to try it and so forth. And he said that when they told him in 2005 he had a third of the state budget, he had like 6 weeks to go before the season started. He said he slept in his car 40 nights in a row. Because if he'd gone home every night to sleep in his own bed, he never could have covered the whole state in that time. But he got the word out. They got 32,000 farmers, 72,000, 100. This is a good example of where some people in the public sector really do, take it upon themselves to provide public service. But he's holding up to this place. This is a conventional plant, 3 plants. This is a single SRI plant. Just look at the difference, not just in the size, but also the color. See how much lighter and brighter color that is. So this fellow is one of our real heroes of SRI. This is in the Indian state of Jharkhand, a tribal farmer. This is some of the poorest farmers in India. And here they have no irrigation. So this is a farmer without irrigation, just using the rainfall. And the average for thousands of farmers now is 7 tons, which almost any irrigated farmer would be happy to get. But this is without irrigation even, when they get these kinds of vigorous plant growth. This is from Iran. Our colleague at the Rice Research Institute in northern Iran sent this picture to show the difference again in the color and size of the roots. Because in fact, this really is the most obvious factor is the difference in the root growth and then the plant health. This is from the Rice Research Station near Najaf in Iraq, sent by an agronomist, a government agronomist again, who took this upon himself to try it and has now had 25 demonstrations across Iraq last year, in spite of all of the conflict and turmoil going on. And you don't need to, these are side-by-side trials, SRI normal. People come look at those and they get the message pretty quickly. This is from Cuba. First cooperative that started using SRI methods back in 2002. This was a 14-ton yield instead of their 5 or 6 they would be getting otherwise. Yep. This is one of the first farmers to use this, and these 2 plants. This is to me one of the iconic pictures, which sort of tells me a lot. This farmer, literally, these 2 plants started in the same nursery. The one on the right was taken out when it was only 9 days old and put wide spacing, single plants, no crowding, a lot of organic matter in the soil, good soil aeration. And then at 52 days, which is when they normally transplant in Cuba, he was sort of going to transplant the one on the left because that's what they normally transplant. And so happens the woman who's our volunteer coordinator in Cuba, a Cornell alumna, by the way, but she graduated 50 years ago. She was there, and she had a camera, so they went and pulled one up from the SRI field to compare the two. And you see there's the 5 tillers on the left, 42 tillers on the right. These are the same genotype, the same genetic potential, but it gets expressed differently. Now, if you look at this picture, you say, well, but you can do a lot of things with pictures. Well, Rena went back the next year with a video camera I gave her, and she recorded for the whole season. So you can go on our webpage and watch this farmer's field the next year where you see these crops growing week by week by week, and you can see how this differentiation comes in, not real time, but in recorded time. This is one of our Cornell alumni working in the Gambia. He was director of this research station before he came to Cornell, went back home, tried it out, 8-ton yield. This is the first farmer in Burkina Faso who tried these methods, had a 7-ton yield, where normally it's 1 to 2 in Burkina Faso. This is a farmer up in Timbuktu on the edge of the Sahara Desert. One of our Cornell alumni, a Swiss woman who's now living in Bamako, got this started last year, and she had an 8.98 ton, almost 9-ton yield on the edge of the Sahara Desert with less water than they normally use for their rice growing there. This year, she said, very excitedly, they have one plant with 160 tillers on it. Now, 10 to 15 is good. One plant, 160. So she hasn't harvested yet, but she's up there right now in Timbuktu and going to send me the results in a couple of weeks for that. This is in Zambia, where normally you get 1 to 2 tons. This fellow volunteer went out and got farmers to try this, 6.144 ton yield. And this is Afghanistan. We're really excited about what's going on in Afghanistan. The Aga Khan Foundation is introducing SRI. So this is not your normal rice growing area. Those who know rice will say that's no place to grow rice. But they're out there planting 11-day seedlings, 30 by 30 spacing. And this was sent to me. A farmer had one plant with 133 tillers on it at 72 days in Afghanistan. And we now have results. He got the calculated yield was 11.56 tons per hectare. Not 2, not 3, but 11.56. But you see that plant, just look how huge and healthy that is. So anyway, now I'm going to get to the theory. I've given you sort of the walk around the world very quickly. This is a case of paradigms, and we're in the midst of a paradigm shift, I would say, in agriculture. And I would describe it as follows. The Green Revolution strategy or paradigm was to first change the genetic potential of the plants so they're more responsive to inputs, to fertilizer, water, etc., and then to give them more inputs. That's how the Green Revolution was made. And it worked. It's been successful. But it's a more gives you more strategy. You only get more if you put in more inputs. The reason it took me 3 years to accept SRI was because I was working with colleagues here at Cornell, and I knew how you did agricultural improvement. You got better varieties, and you put more inputs. And SRI didn't do either one of those. We took any variety, old, new, traditional, land-raised, whatever, and we reduced the inputs. Now, there was more labor initially, more knowledge, more skill involved, but not material inputs. And so SRI, or what I call agroecology, is the general area. Instead of changing genetic potential of the plants and adding more external inputs, we changed the management of the plants, the soil, the water, and the nutrients, first to promote the growth of big, healthy, functioning root systems. And you've seen some pictures of that. And what you can see then is that we also increase the abundance and diversity of soil organisms, better to enlist their benefits. And if we do those two, the things that cause those two things, we then get a better phenotype, healthy or productive. Do you all know what a phenotype is? Or who knows what a phenotype is? Let me put it that way. Shailesh does. Anyone else? Remember your biology, I hope? You all took biology, I hope? A genotype is a genetic potential that all organisms start with. The phenotype is the phenomenon that eventually results as the genetic potential interacts with the environment. We talk about G times E. The interaction of genetic potential and environment gives you a phenomenon, a phenotype. And so what SRI is doing is not changing the G, which is what the Green Revolution did, we changed the E. And it turns out you can do an awful lot of good things at very low cost and very quickly by changing the E, giving a more productive environment. This is, we were told by the leading rice scientists in the US, who I asked to join us, is you can't study something with 6 different variables, because doing factorial trials where you take all combinations of 6 variables gets you, and then replications gets humongous numbers. So he walked away from it. He published an article 4 years later calling this a voodoo science, by the way. But we got students at the University of Tananarive in the ag school, they're honor students, to do their theses using factorial design with 6 different factors. One had 288 trials on farmers' fields, 2.5 by 2.5 square meters, random block design, 6 replications. Huge job, but he's also very brilliant. And another did 240 trials at another location. One was at sea level, sandy soils, tropical environment. The other was 1,200 meters, temperate environment, better soils, same pattern. So you see here that the local variety on clay soil, the better soil would like that, it went from conventional practice to 1/4 to 1/2 to 3/4 to all SRI. Local varieties on loam soil, not as good soil, the same pattern, though not as high yields. A high-yielding variety on poor soils is this red line here. See that? And then local variety on sandy soils is the least good of them all. But we saw this from student research, that this is the interaction among these practices. And this wasn't including also active soil aeration yet. So this wasn't a full test, but it was enough to show that we were getting some really remarkable results. And that's a 10.35 ton yield there with replicated trials in a country where the average is usually 2. Anyway, this is a master's thesis done here at Cornell, doing a regression relationship between nitrogen uptake and the grain yield for SRI and conventional methods. The light purple is conventional methods. And as you add more nitrogen, or the plant takes up more nitrogen, you see it goes up and it sort of plateaus somewhere 4 to 5 tons because it doesn't have good roots. It's crowded together. The tiller isn't so great. The SRI plants on the same fields or with the same farmers, side-by-side plots, you see the different response to these practices. So we're seeing a very different change in the phenotype, the phenomenon that emerges from the same genotype, genetic potential. So,
where did the nitrogen come from?
From the soil.
It wasn't added.
No. It was added if most of the farmers didn't even put on compost. So most of them were doing actually with whatever was in the soil already.
Okay.
Yeah. But now that I'm just about through here. SRI, I want to emphasize, is a work in progress. We expect many more changes and improvements to be made, especially from farmers. This has been an innovation where farmers have made a lot of the advances because we present this to them not as a technology to adopt, but a set of ideas, principles, observations, and ask them, make your own adjustments, your own adaptations, and, if possible, improvements. And I'll show you a couple examples. We're spreading SRI, but we regard farmers as partners. We don't like to use the word adoption. They're not adopters. We want them to be partners in capitalizing upon this opportunity, which SRI offers. We say it's to be adapted, not adopted. It's not a technology. It's not something finished. A set of practice, rather principles, concepts, and it's not yet finished. And we're now applying it to rain-fed areas, to other crops. This is an example of a farmer innovation, where in order to aerate the soil and plow up the weeds, he made this for $3 of materials, and those things sticking up are nails. They drove into a wooden axle. He got a blacksmith to weld a frame. His first model didn't have this rake on back. He did that in his second model because get more soil aeration that way. And he said he got $20 worth of rice more. He's got a small field, not even as big as this auditorium. But he's got $20 worth more of rice for his $3 worth of investment in materials and about 75 cents worth of labor. He's calculating pretty carefully. This is surprising. This farmer, first time he tried SRI in the Terai of Nepal, the weeds killed him. I mean, he didn't get a very good result. His neighbors thought he'd never try it again, but he didn't. He went back and thought about it over the winter. And what he designed is something that looks like a broom, really. And he said it cost him 25 cents to make that. And he drove nails into this. You turn it upside down, and then you go up and down the rows. You can dig out the weeds that way, and you aerate the soil. And he says, one person can weed as much area as 2 or 3 laborers going through pulling my hand anyway. So he says labor and gets a better result, double yield for this little 25-cent implement that he has developed. This is the best weeder. This is a metal one, designed or redesigned by a farmer in Sri Lanka. It costs about $10, but it's really a very practical, very effective one. And this is also in Sri Lanka where a farmer put a motor on a weeder so he could actually have some power assistance. And he says he can do 2 hectares in 1 day, all by himself with his power. What first got me interested in this is that this is from back 97, 98, when we were still just getting started in Madagascar. We had 2 farmers did no mechanical weedings, and they got almost 6-ton yield, which is more than double usual, but still only that. The 35 farmers did 1 or 2 weedings, got 7.5 tons. The 24 farmers who did 3 weedings got a little over 9 tons. The 15 farmers who did 4 weedings were over 11, almost 12 tons. Now, this is what really sort of struck me. This is not fertilizer. This is simply soil aeration. And what happens if you aerate the soil? All those soil organisms that require oxygen can multiply. And it turns out they do all kinds of good things. That's what I've been learning in the last 10 years, is how to understand soil biology and the contributions it can make to agriculture. This is from Nepal, where we had 412 farmers who 32 of them did only 1 weeding. That's the minimum. That was their average yield. 366 did our recommended 2 weedings. That was their average yield. Normally they get 2 to 3 tons here, so this is like double. But the 14 farmers did 3 weedings, got 2 tons more yield. Now 2 tons, that's a huge amount for farmers to get that as a bonus, because there's no other cost than just the weeding, which is their own labor. This is a picture from India of sugarcane, where normally yields are about 65 tons per hectare. Farmers using these ideas now with sugarcane say they're getting 125 to 235 tons per hectare. Because these canes will weigh about 1 kilogram each. These will weigh 2.5 to 3 kilograms. So you cut the plant population down, but then you get bigger, healthier, more productive canes. This is finger millet, a very important crop for the poor in India and much of Africa. On the right you have a traditional variety and traditional management. Improved variety with traditional management, that's improved variety. They call it SFM, a system of finger millet intensification. But again, look at the phenotype difference that we can get by giving the plant a different growing environment. So anyway, what's going on to produce these phenotypes? In addition to the root growth, which you can see, you get the abundance and diversity of soil biota, soil organisms you cannot see, except for the earthworms, or some of the mites and springtails and stuff like that. We get nitrogen fixation by nitrogen-fixing bacteria. We get phosphorus solubilization by what are called phosphobacteria that get the phosphorus from unavailable portions of soil for themselves. And then when you flood the soil, they die, they burst and release their phosphorus into the soil solution so the plant can get it. We get nutrient cycling by protozoa and nematodes. Do you think these are, protozoa and nematodes should strike fear into your hearts because they give all kinds of terrible things, dysentery and so forth. But it turns out that they are cycling nitrogen from the soil, from the bacteria that live on the earth's roots, or the plant's roots, so it's made available to the plant. 20 to 40% of the nitrogen that plants get is cycled by nematodes and protozoa in the soil system, if it's a healthy system. If it's not, they don't get that benefit. We have water and nutrient access through mycorrhizal fungi. These are funguses that infect the roots and spread out little filaments into the soil and acquire phosphorus and micronutrients and water for the plant that they share with them. If you flood the soil because these require oxygen, you don't have any mycorrhizal fungi. So one reason why not flooding your rice plant gives you a boost is the plant has more access to nutrients. If you have these fungal infections of the roots, these are beneficial infections. Not all infections are bad. In fact, a lot of them are very good, as I'm going to say in a minute. Phytohormones. The bacteria are producing auxins and cytokinins and gibberellins, other hormones that in fact stimulate the plant growth of roots and the plant performance generally. We have something called induced systemic resistance, where plants are more immune to pathogens when there are certain bacteria living in the root zone with them than when they don't. So we've done controlled experiments, not we, scientists have done controlled experiments showing that the presence of certain bacteria in the root zone gives the plants more immunity to damage from pathogens. This is research, again, that helped me understand what was going on, done by a student at the University of Antananarivo in Madagascar, and doing two different kinds of soil. In the clay soil, if you had traditional cultivation with no fertilization, no soil amendments, yield was 1.8 tons. If you simply did SRI methods but still no soil amendments, you got 6.1 tons. So by young seedlings, wide spacing, one plant per hill, aerated soil. If you added nitrogen, phosphorus, and potassium, you went to 9 tons, about 50% more. But if you instead you put compost instead of fertilizer, you got 10.5 tons. What was going on? They looked at the bacterium called Azospirillum in the roots. And you have 65,000 here, 1.1 million here. If you put on NPK fertilizer, it goes back by 60% because that inhibits the production of the enzymes needed for producing nitrogen. But then if you go to compost, you get 1.4 million. So you see that there's a relationship here. Loam soil is a different structure. He just did 2 of these because these are all replicated trials, a lot of work. But you had, if you just use the methods on this soil without any soil amendments, you got 75,000 and a 2.1 ton yield. But with compost, this soil just magnifies. So we're seeing that the way you manage the plant, the soil, the waters, and nutrients affects the inhabiting of the roots by these bacteria, and it has a very pronounced effect both on the number of tillers per plant and on the yield. And this is really exciting. This will expand your minds a bit. Research just recently from China where they looked at how soil bacteria, rhizobia we call them, infiltrate the roots, go up the roots, up the stem, into the leaves, into the leaf, called phyllosphere, the leaf sphere. And they did controlled experiments where you had controls, but then you have 5 different kinds of rhizobia that were allowed to infect the plant, not just the plant, but actually the leaves. And it turned out that the grain yield per pot, instead of 51 grams per pot, it was 77, 64, 61, 86, 86. And you saw more photosynthesis, more water efficiency. Somehow the infection of the leaves by soil bacteria changed the physiology and the functioning and the productivity of the plant. More chlorophyll, more photosynthesis, and more yield as a result of the infection, quote unquote, of the leaves by soil bacteria. This has never been looked at before. They had to use electron microscopes to track this down, but it's published in the Journal of Applied Environmental Microbiology. Michigan State Professor Frank Dazzo is one of the advisors for. He's a co-author for this. This is really good, most modern science, understanding the relationship between plants. By the way, when you came up in the elevator, you notice there's a new name for the department in this building. It used to be called plant pathology. What is it called now? Plant pathology and plant microbial interaction, something like that. Plant microbial biology. So whereas we always, when we talked about microbes before, they were pathogens, bad guys. And we said how to get rid of them, how to control them. But in the last 10 and 20 years, we've discovered that the interaction between plants and microbes can be very, now it's the plant pathology and plant microbial biology. So here's my last slide. Lesson for all of us. We are not alone. Plants and animals, including humans, have co-evolved with microbes for millions of years. And we just didn't pay much attention because mostly we thought about the bad guys who made us sick, not the services and benefits of all the good guys out there. How many of you have seen the article, probably about 6 months ago, saying that 90% of the cells in our bodies are not our cells? You missed that? It was published in Nature and lots of pickup on that. You have almost 10 times as many microbes in your body as you have cells of your whole body. Now, they only add up to about 2 kilograms of weight because they're very small. But you are not just Homo sapiens. You are a system which interacts with and depends upon. There are certain vitamins that you depend on for your life which you cannot synthesize, which only these microbes can produce for us. There's certain protection against pathogens, which we're very vulnerable. Even asthma now, we're discovering that if you protect people from make it a dust-free, dirt-free environment, they don't develop the immunity to be able to resist the effects of asthma. There are all kinds of ways. And with both plants and animals, are thoroughly interdependent on the services, on the cohabitation of microbes. So what I want to say is my prediction is that the productivity and sustainability in the 21st century will depend on a full and proper understanding of its microbial realm, which we are only now beginning to get a decent handle on. I would encourage you, do you all know the name Lynn Margulis? You don't know she was Carl Sagan's first wife. You all know Carl Sagan. They had 3 years they divorced, but she's brilliant. She's a microbiologist at Amherst. And she's a paperback. She's written lots and lots of books. I've read everything she's written. She's one of my main sources of knowledge about microbial work. But get her book called Symbiotic Planet and read it. Because I think we're not going to be able to get through the 21st century with our very outmoded notions that here we are, we human beings we're at the top of the evolutionary ladder. Her book is about how we will decenter Homo sapiens from being the top of a ladder to being part of this, vast interconnected web of plant, soil, and microbial organisms, which have to live together or they probably won't live at all. You may remember that when you took a biology course sometime back, there was this tree of life, which started with microbes and archaea and fungi, and plants go off and animals go off. Remember that one? Yes. That's good taxonomy, but lousy biology. The fact is we never separated. So both plants and animals, we being animals, are still continuously interactive with and dependent on microbes. If you live in a germ-free environment, you die. Now, there's some bad guys and some really bad guys. I'm not saying you should love them all. But in fact, there's a study in Nature magazine about 3 months ago. 95% of the organisms in our body are permanent and benign. The 5% that are pathogens, they're transient. They come and they go. But most of the inhabitants, the residents, are ones we need. Or sometimes they're neutral, but then they're beneficial for other organisms. So it's a really complex world. And so in that sense, what I'm leading from my rice start, showing also how I've evolved in my thinking over the last 10 to 12 years, not because I was interested in science, not because I want to be a proponent of biology, because I want to help feed the world. That's what I've done for the last almost 40 years, worked on food and hunger issues. This rice system opens up some really interesting opportunities to overcome the constraints we presently have, certainly to meet the basic food needs of people. But let's not stop there. First of all, we don't stop with rice. We want to other crops, sugarcane, system of wheat intensification, both Himachal Pradesh and Uttar Pradesh. Now we have wheat being produced with these SRI methods, getting, according to the latest article, 3 times more yield of wheat using these concepts for wheat. So what it's doing is kind of changing the paradigm from a system where we look at plants as little machines. And we design them. We do our modification, our breeding, and so forth to get a better design. And then we fuel them with our inputs, make them do what we want. This vision I'm talking about here is one where really, we understand the plants are organisms. They have their own evolved history and capabilities, their own interdependences. They exist in this welter of all kinds of organisms, rather than being something we pick up here and we put there, where we're totally in control versus us being sort of related to and with them. Father de Launier, in his only published article at the end said, I just love it. It was in French, but he said, the rice plant is my teacher, mon maître, my master. I learned from the rice plant. And what he did was simply spend his time observing, testing, trying to learn from the rice plant. And this is what he was able to actually synthesize. And we're finding out things far beyond what he could even know in his 75 years of life. And I think this is an exciting time to be alive. Some of you may know the movie The Graduate, where, plastics, young man, plastics, I'm telling you, microbes. Young men, young women, microbes. I think that in the 21st century, we're going to make it through, if we make it through, in part by really getting in touch with, in sync with the microbial world. And that's I say, but at least to be an educated person in the 21st century, you've got to read something like Lynn Margulis' book, Symbiotic Planet. You've got to understand that. and stop thinking, we're in charge and we're in control of everything. And we need to have a very profoundly ecological understanding of ourselves and everything around. Anyway, let me stop there. And I'd be glad to take questions for as long as you want to stay around, but you'll have your limits. We break at 4:30, is that right? Or what time do we break?
We can continue and take some questions.
Okay.
Neil will moderate.
You want to be the moderator, please?
Sure.
Yeah. Who's first?
All right. Leah had questions? Please, we wanted.
I'm interested in the question of how agricultural research links to agricultural research or lack of enough of emphasis on agricultural research links to the current crisis of food prices trends, and how do you see that as being able to address the issue?
Well, as I said before, the Green Revolution, which was a product of scientific research in the 60s and 70s and 80s, made a very substantial and important contribution to world food supply. Since about the mid-90s, the Green Revolution has been running out of steam. The rate of increase has been heading off and even getting fairly stagnant, in some countries even dipping a bit. And I think we sort of reached the limits of how far we can go by changing the genetic potential and relying upon an input-driven strategy. So it's very convenient that SRI insights are coming along at this time. I'm disappointed by the fact that this has been really resisted by many people in the agricultural research community. This is a different paradigm. And I say, I can understand skepticism, because I was skeptical for 3 years. It took me 3 years to accept this. Not until 97 did I finally say, well, okay, I'm going to start trying to get others to try it as well. But I guess, what I would say is, skepticism is very good, but it's something to be optimized, not maximized. And some people sort of they have their strategy to maximize skepticism. And that's not the thing. Optimize it. It's a good stance, but not to be taken to extremes. And we've encountered some real resistance, which is simply looks at our data and says, well, it can't be true because our models show that you can't get those high yields. And I see, but if you have models, that's fine, but let's still look at empirical reality. And so I guess I'd like to see an agricultural science which is really still based on empirical reality, which doesn't assume that we know everything yet. Again, if we did, it'd be really surprising. We know all previous generations have not known everything. Certainly our parents didn't know everything, and our grandparents certainly didn't know everything, but now do we know everything? And so I say we should do the best we can as scientists to construct the best explanation we can with most reliable data, most creative theories. But let's never assume that we know everything yet, that we got it all right. And I was benefited by a father who did biochemistry undergraduate degree back in the dark ages, even before the Depression. But he told me in high school when I was doing chemistry, he said, just remember, Norman, that almost everything I learned at the University of Wisconsin in my chemistry courses back in the late 20s, early 30s is now obsolete. And he said, what you're learning now will, in similar time, probably be obsolete as well. That didn't mean I shouldn't learn it. Didn't mean I shouldn't respect it. But it didn't mean I should be open to the fact that it's not the last word. And so I make that as a general proposition for all of us in all of our disciplines. And all scientists should be genuinely open to the possibility that maybe we don't know everything yet, and there will be some new ideas coming up. But I hope you've all read the book by Thomas Kuhn called The Structure of Scientific Revolution, which talks about paradigm shift. No one should go through graduate school without reading that book. I did it in mine, but it's still relevant today. He talked about how in our sciences, all disciplines, we had these paradigm shifts, and then all the sort of struggle, the blood on the floor, the bruised egos that comes with that. Let's be prepared there are going to be paradigm shifts. That doesn't mean run after every new idea. Probably a majority of the new ideas are still wrong. But how do we keep ourselves open to that and always look for empirical testing and validation? So I think that agricultural science, partly because on the soil biology front, there's a lot of new stuff coming up, and it's really very exciting. Some of you might have gone to the seminar Dan Buckley gave this noon in Crop and Soil Sciences on his work on soil ecology. Fascinating work. Difficult. But he's the start of what's going to be a 30-year career, getting to understand some of the ecology of the soil organisms and how they vary. So I think we've done a lot with agricultural research. I think that investing more in the present paradigm isn't going to get us very much. And unfortunately, we have a lot of sunk investments, you might say, in the present ways of doing things and researching them. I think we need to be prepared to shift. And so I'm trying to encourage people. Now, you're not agricultural scientists, but you are a person who will have some voice, I hope, in how we use resources and set priorities, or how we just keep our minds open. And for that, all the help you can give is welcome.
Okay, behind, yes. I'm sorry.
Professor Albert, I was curious about the effectiveness of SRI vis-a-vis high altitude areas. The picture from Afghanistan seemed like it may have been
Right.
high altitude.
In Nepal, where we have the most experience with high altitudes, we've had it, about 600 meters. No, well, the Terai is maybe just about 200 meters. It's not very high.
No, it's not.
Not even that. Up to 2,700 meters, we've been able to see the effect. So that's pretty high up. So we don't find climate, higher altitudes are not as productive as warmer, lower ones. But we do see that if, field by field, you get better results with the young plants, wider spacing, soil aeration, et cetera, even up to 2,700 meters elevation. So that's not a constraint. I think the one constraint, you're asked a question, where can't you do this? If you have continuously inundated soils because of monsoons or lack of drainage or whatever, if you can't create aerobic soil conditions, you won't see this effect. Now, you might still get some benefit from some of the practices, but you won't see the kinds of dramatic improvements that we can see when you can control all these factors and create that growing environment.
Professor, assuming that people accept this paradigm shift that you're talking about with the SRI, how transitionable is it to these methods to like industrial farming applications?
Good question. We've had in India operations as much as 40 hectares, which is 100 acres, with an 11 plus ton harvested yield, which is really good. There are several operations which can be mechanized here, which I think will be done. If I have only 1 hectare or half a hectare, I will do transplanting, very labor intensive, to get the maximum benefit from my little piece of land. If I have 2, 3, 4, 5 hectares, I'll do a method which in Sri Lanka, from a farmer, is a very interesting method. What he does is he takes germinated seed, prepares his muddy field, then he broadcasts it, just throws it out, but 5 times as much per hectare as we would do if we transplanted. And then at 10 days, when the plants are just coming up, he takes his weeder and he weeds in a lattice shape, and he eliminates 80% of the plants. So there's no nursery, which takes a lot of time, no transplanting, which takes time. We have a professor visiting from Tamil Nadu Agricultural University right now, this month, who's done a study of this, and he found 40% labor saving that way without this. Now, he and I are talking how to do the next step, which is to have weeders that are mechanized. You all know these little sort of 2-wheel Japanese tractors. At least you've seen pictures of them. And he and I are sure that you could design a weeder implement which you put on the back of them, so you could actually weed 3, 4 rows at a time with a little power tractor which could go very quickly, in which case the sowing could be even done from airplanes, I suppose. And you could actually then bring in, and the weeding would be even better because you can aerate the soil better with more power behind it than just the human power. So I think there will be further, remember, this is not a technology, it's some insights going to the work of Father Lalanier, which we're still learning more about all the time. So I think we could actually take these principles and take them up to larger scale. Now, I guess in California, if we're planting from airplanes on 10,000 hectare plots, you could try to adapt. I'm not as confident. For me, again, the big problem for the 21st century is still going to be how do we help the smaller farmers where most of the need, most of the hunger is. But because this is in the biology of the plant, not anything we control or own, there are no IPRs, no patents and stuff to control it. This is going to be adapted on a large scale. And I think it's going to go into other crops, not just rice, which will be very interesting.
Yes.
What is the major reason for resistance from the agricultural community here at Cornell? What is the reason behind it? Is there a downside, a bad downside to this?
I look for it because I sound very optimistic, but I also have a very skeptical and critical side, as some of the students here know. You know, I'm not an optimistic pushover, am I, Shailesh? No. I haven't, the one thing, their constraint is, first of all, water control, if you need an aerobic environment. For many farmers, learning is tedious and labor-consuming, but we're finding that, like in Cambodia, they save labor the first year, but it takes kind of a mental mindset shift. So in the first case, it's a mindset for the farmers. It's also a mindset then, I think, for the scientists, and that this really shifts the paradigm, which I set up there. You know, you got to get better genes, and we have a huge investment of time and resources and equipment in the breeding approach to getting yields up. And then secondly, there are a lot of interests, you know, in implements, in fertilizer, hybrid seeds, etc. I think see this as, if not threatening, at least sort of an insult. The other thing which I can't quantify and I can't prove is the fact that I'm a political scientist doesn't help. I don't have any credentials for this. Now, I think I've learned a lot in the last 10 years, and I can, in the book which you mentioned, you know, we've got some of the best scientists in the world contributing. I edited it, but I learned that way. And we got a lot of scientists who understand this, and they got their own evidence to support these kinds of conclusions. I think partly it's just, you know, human nature and inertia. And my colleagues here at Cornell have known me as a political scientist all these years. One of my best friends, who's a very innovative guy, still growls to one of his students and my students, what's this Uphoff, you know, political science doing in rice? So like, what the hell can you know about rice? And when I started, I didn't know much about rice, you know. But I can learn, and that's a lesson for all of you too. You don't have to stay, if you can think well, if you can respect evidence, if you can, you know, find people whose, you know, views are worth, and by the way, we have a lot of scientists outside of the National Rice Research Institute, outside of Cornell, who've been doing this work, China, India, Indonesia, who are excellent scientists. I mean, I wouldn't be as bold to say this if I didn't have scientists who have the qualifications, who've done the work and reached the conclusion which I'm reporting. But this is complicated. I don't have a full answer for this yet, and this is not through it anyway. If someone wants to study this is an interesting subject. You know, we'll be glad to share all our information, all the emails and everything else, try to make some sense out of all this. But I'm sure it's not unique either. I mean, this is typical for a lot of innovations. My neighbor up the hill, Dr. Lempert, some of you might know, he's an ophthalmologist. And, you know, he developed a method for measuring the diameter of the optic nerve with sort of lasers. And he discovered that amblyopia, which is a big problem of, you know, where the eyes don't track, which for the last 50 years have been said to be an abnormality which if you patch an eye and force the other eye to start tracking, you can correct it. And it so happens about 60% of, you know, pediatric ophthalmologists are working on this problem. And he said, no, he's documented with hundreds of cases that amblyopia has a physical base in terms of the formation and diameter of the optic nerve. So it wasn't so much behavioral as it was straight physiology. He had 10 years of resistance on this, you know, being kept off panels at meetings. And 2 years ago, he finally had an editorial in the Journal of Pediatric Ophthalmology apologizing for, you know, this. But the resistance came more from people at Harvard and Johns Hopkins, all the prestigious schools. What can this local medical doctor know about these kinds of things? It happens he's really brilliant. He had the evidence based upon an innovative measurement device. And now it's sort of coming into literature. And so he and I would commiserate. You know, we have the same kind of problem. He's in ophthalmology, I'm in, you know, sort of agronomy. Is this kind of resistance, but often from the most established and prestigious places. So again, I make this comment because you should be aware that this is a bias, I think. And Cornell happens to be among the elite places. We in the elite institutions think we've really got it right. After all, if we weren't right, we wouldn't be here. Right? So there is a problem. You know, look for new ideas sort of off the reservation sometimes. Or be open to new ideas that come from unexpected places. You see, part of my problem has been one of my colleagues, I won't mention her, but she says, you're not a scientist. That was the only answer she'd give when I started giving her data from, you know, some brief, you know, you're not a scientist. She says, when it's in the published literature, I'll accept it. Until you get in the published peer-reviewed literature, it's not there. And that's the idea. If the tree falls, but it's not in the published literature, it didn't fall. That's not true. Not.
Use the published literature.
Well, we're getting into it now, but the point is, not everything that's true is in the published literature, And conversely, not everything that's in the literature is true. If it was, we could give up. Once we got it, we don't need any more. We keep a literature, so we continually try to improve upon and expand what we thought we knew. So again, my point is keep, skepticism, but optimize, not maximize it. Be willing to look or accept, at least consider evidence from unconventional sources, like a priest in Madagascar. Now, it so happens he's a really brilliant person, had a lifetime working on this. It's like Gregor Mendel gave us the science of genetics without any training in biology, right? He was a good observer, very systematic, and he laid the basis for modern genetics as a priest. Or a monk, I guess. He was not a priest, but a monk. But the point is, how do we keep our minds open to these unconventional sources? And always keep telling ourselves, remember, we don't know it all yet. When I go into a village, I always try to remember to tell myself, Norman, remember there are going to be some farmers in that village who are probably smarter than you are. I can have a PhD from Berkeley. I can be a professor at Cornell. But that doesn't mean I'm the smartest person in the world. And it helps me do my rural development business if I keep reminding myself. And I can't, maybe there isn't. I'm saying I should always be thinking there could well be people that are smarter than I. And we should do that again in all of our work. Whatever profession we're in, keep in mind the intelligence is very broadly and often unexpectedly distributed. But how do we make the best of whatever we have and in consultation and cooperation with others? The problems we face, especially in the 21st century, can be so complicated that none of us can expect we're going to, by ourselves, make that much impact on them. We've got to figure out how do we build on others' knowledge and their intelligence, their experience, and make it a collaborative effort. Anyway, I'm getting very philosophical. So, any more questions? Yes, back there, there's one. Speak loudly, okay? Mm-hmm. Well, it's interesting that one thing we hadn't counted on originally is that when you harvest the unhusked paddy and then you mill it, we usually get about 15% more polished rice per bushel of unmilled rice because there's less unfilled grains, so there's less chaff. And there's less shattering during the milling process, so fewer broken grains. So actually, there's a bonus of 15% beyond what I showed you up there, which is the paddy unmilled rice harvests that in Cuba, in Sri Lanka, China, India, we all have evidence about 15% more edible rice per bushel. In fact, I remember in Sri Lanka, I first learned about this when the ag official said, millers are coming to farmers doing SRI in the field before they harvest and offering them 10% more per bushel of paddy. And I remember thinking to myself, I don't need any data. If millers will come and pay 10% more per bushel, they've got to be getting more than 10% harvesting because millers don't lose money. They're some of the most self-interested people in the world. So, but we've got data from China. When I was in villages in Tripura, state of India last October, I was asking, what do you get back when you mill your SRI paddy? And it worked out to be 18% average return. So that's actually another contribution to world food. That farmers say that they like the taste and the smell of SRI paddy better. It's more attractive. Farmers in Sri Lanka said, if we cook it at night, it's still good in the morning. It doesn't get all mushy like some of the new varieties do. So we think there are some advantages in the quality of the grain. We think there's probably some nutritional advantage because you get more micronutrients if you have a bigger, deeper root system. The fact it doesn't shatter suggests there's more protein content as well because that's documented in the literature that you get less shattering if there's a higher protein content. But a lot of things I can't answer because we don't really know yet. There are lots of things still to be known and discovered. This is a work in progress. And I hope, 3 or 4 or 5 years from now, we can say a lot more things and say them with more confidence than what we see now. But the amazing thing is, so far, we haven't found downside. You know, I'm willing to accept it. And we got so many good things anyway. You know, we can easily accept some bad things. But we just haven't come across them. We keep coming up with good, favorable things. Biodiversity. It turns out that traditional varieties can give you 6, 8, 10, 12-ton yields. Now, the high-yielding varieties, the modern varieties, give you even more yield, but consumers like the taste of the traditional ones better and will pay 2 to 3 times more per kilo. So a farmer can get more income by producing, you know, the old varieties. So we're hoping we can help conserve rice biodiversity by making it profitable for farmers to grow the old traditional heirloom varieties, which are really marvelous. There's some marvelous things out there. I've had lots of them. Some of you may already know about them.
Okay, then, do we have any other questions? We have time for maybe one more.
Anyone got a last one? Nothing burning? I'll hang around if anyone wants to, you know, come shoot me a question individually. You see, this is really, you know, let me just have kind of a peroration. You know, the 21st century, which all of you are entering in, which you're going to see much more of than I will, is not going to be as promising as the future I could look forward to when I was your ages, when I finished my MPA. You know, there's such optimism and development. We're going to be able to, you know, get compound economic growth and all that. You know, my daughter, who was in SEPA for a while and worked with David in CRP, did her thesis, not on development, but on post-war reconstruction. How do you put places back together again after they've, you know, gone into the gutter? And the fact is, the world that you have to deal with has got a lot more, you know, negative prospects to deal with. I'm sorry, I'm not personally responsible for something in a very small way, maybe, but the fact is that, you know, you've really got to then be smarter than we were. You know, you got to think more creatively and be prepared for some new things. We got to get out of the ruts we're in. We're in a lot of ruts right now, and but on the other hand, at least in the area of food, I think we can actually solve our food problems fairly swiftly in the next 5 to 10 years. And again, that won't solve all the world's problems, but it will, if we can't solve those, the rest of them are going to be really difficult. So my hope is at least some people will spend the time making sure that we get that food basis solidly and securely and sustainably laid. I think, though, we can see how it could be done. And that's why I'm spending most of my remaining time trying to get this better understood, better spread out, and diversified, ramified everywhere we can, and go beyond rights to think about postmodern agriculture. How do we remake the agricultural sector? And that's a big challenge. And again, I won't do it myself, so if anyone's interested in that, you know, here's a really good thing to work on. It's not the only thing. But again, if we don't do that right, everything else is going to be really hard to do. So anyway, thank you for paying attention. I was going to do this next semester, but we sort of moved the time up. So I'm always glad to tell you anytime, any place.